Another component, major component of food are proteins. Like carbohydrates, these are going to be a combination of much simpler molecules. Also, like carbohydrates, these are going to allow us to store energy and also do some work. But unlike carbohydrates, these are much more involved in the processes of life. So the proteins are involved within the cells of all kinds, and they are doing the work. They're actively involved. Things are built out of proteins. Proteins are used to send signals, do work, all of which is involved in proteins. Like carbohydrates also, though, we get about 4 calories of energy for every gram of protein we ingest. So we can still get a substantial amount of energy by breaking these down into their parent things. Again, they're polymers or long chains of things we call amino acids. So here's 2 example amino acid. We have glutamic acid on top and cysteine on the bottom. There are several other amino acids that are common. I just want to give you representative samples. Again, you see that we get a large amount of bonding that's able to happen on the edges here. Just like in the sugars, we also are able to get intermolecular interactions. This hydrogen bonding is capable here, so we can get various sort of structures. When we react these amino acids together, we have one thing that's called amino acid. Put a couple of them together, we call it a peptide. Put a whole bunch of them together, we call it a protein. When we get these long polymers or long chains of amino acids, they become very complex, and we can describe their structure based on these 4 different levels. The first level, or the primary structure, is determined on which amino acids are bound in which. So if we start on one end of the molecule, we can then label each of the amino acids as we go down this long polymer. And in this, then, that is the primary structure. So we can say that there's glutamine, a glutamic acid next to a cysteine, next to other amino acids as we go down the line. That's going to describe sort of the basic structure of this protein. The next layer is, because all of these are capable of interacting with each other because of hydrogen bonding, the sulfurs here, we can get sulfur bridges. We're going to allow these long chains to start forming more complicated structures. The most common of which is the helix that we see, the DNA, which has that double helix, that sort of spiral that goes up. That is a result of this cross-linking, this secondary structure. Another common secondary structure in proteins is called a beta sheet. It's much more linear, where the helix sort of spirals up and becomes sort of a cylinder. The beta sheet becomes just like that, a sheet. It goes up and down, sort of a wavy sheet that's fairly long in length. So these 2 result from the intermolecular, the parts of the molecule interacting with each other. Then we can take this sort of now long protein that has these various shapes, and we can combine them over a distance. So we can see a protein that is going to start to develop a 3-dimensional shape overall. So we could have some alpha helixes or helixes in there. We can have some beta sheets to give us sort of a flat zone, some sort of circular zone. So we can get a 3-dimensional shape. This is called the tertiary. It results from those shapes folding together. The last one is this 4th degree, which relates to this protein molecule interacting with another protein molecule. So we can take another different one and we can actually form them together. They can connect together to form something of much more complexity. And we can think about this in terms of like an enzyme, which is a protein. Enzymes cause reactions to occur. They're catalysts. So we can see if these 2 proteins come together to form a pocket, if we get a molecule that fits into that pocket, once that binds, that's going to change the shape and allow some other process to occur. So this folding is going to allow for very complex sort of chemical processes to occur within a cell. But because this is a very complex nature and it relies on some of these interaction between molecules, it's very easy for those processes to be disruptive. When this folding process is disrupted, we call that denaturing. That protein sort of falls apart, loses its nature. And it's fairly simple to do for these proteins with a little bit of heat. We can do it sometimes with light. We can do it with changing the salt concentration. We can do it by changing the pH, all of which is going to affect the conditions that these molecules have and cause them to either change their folding shapes or sort of unfold completely. And we're going to take advantage of this denaturing in cooking. It's going to change the textures and the feels, allow some new bridging to occur. All of this process is by exposure to light, heat, salt, acid. All of this is going to affect the texture. If we look at proteins in like meat, for example, when we expose it to high heat for a long time, that's going to break down those long, narrow muscle fibers and the things that hold those muscle fibers together and allow them to sort of become fluid and move around, which is going to allow us to have a nice, tender piece of meat as opposed to a fairly compact, tightly held, chewy piece of meat. All of this is going to play a role. We're not going to spend a whole lot more time talking about meat protein. We're going to spend some time talking about proteins in other areas. The first of which is in eggs. So if we look at our nice diagram of an egg, we all have had eggs before. We have a yolk inside of this shell, and then the rest of it is surrounded by what we would call the egg white. The yolk does have some protein in it, but the majority of the components of a yolk are going to be those fats and other things. So we're going to sort of ignore that for now. We'll come back and talk about those. Although we'll see when we make mayonnaise that those are going to have some charged particles, so we can get some emulsifiers out of the yolk. But right now, let's talk about the proteins that are available in this egg white. Right, when you open a uncooked egg, we see that the egg white isn't actually white at all. It's a sort of viscous, sort of thick, clear, solution. It's able to separate fairly easily from this yolk. That egg white has these proteins within them. This is a short list of all the proteins that are listed in egg whites. Things like albumin, which is the major component of the egg white. It's actually where we get the egg white from. When we expose this to heat or salt, it denatures and we get that nice white texture that we're seeing in the egg white. We also have some globulins, which are going to allow us to make transitions between fats and these proteins. We have some enzymes. This is required. This lycozyme is required in once that egg starts to develop. If we don't eat it, it gets fertilized and becomes a new chicken or whatever. That's involved in breaking down some of those processes to get that egg to survive. We also see some of these others, but these are a wide range of different things. We also see proteins present in milk. And we know milk contains two different things. We have curds and whey. The curds, while having some proteins in them, like the egg yolk, are going to be mostly the fats. And we get the curds, we collect them, and we make cheese, which we'll do later. But inside the whey, we have, so if we look at milk here, we have these small pockets of fat, which are going to be related to like an emulsion. So we have these fat in this liquid solution. We also have then two different types of proteins. We have this casein proteins, which are going to form micelles, which are sort of small bunches of these proteins. And this is in the casein micelles is where we're actually going to have the calcium B trap that we're so familiar with in milk. We also have the whey proteins, which are smaller. They're going to be much less complex, all built in this sort of larger emulsion of milk. Again, just like before, this is a very straightforward, sort of short overview of proteins in food. There's a lot more involved in the reading and in the activities. Let me know if you have any questions.